know what it's supposed to first. Okay. All right, guys. Okay. So, you got your test coming up next time. It's going to be over chemistry and biochemistry. Okay. You've got the study guide. Get that done. There will be another test coming up shortly. It will be after that. It will be over cells, right? So when I say cells, I'm talking membranes. I'm talking organelles. I'm talking basic cell functions, the stuff that we've already started. It's going to be a vocabulary-rich kind of a time. I can tell you more about what that test is going to look like even than I can about this one. You're going to have to label things, or at least if I gave you a diagram and I gave it, you're going to have to be able to point out and tell me, oh, that's centriole, that's a nucleus, that's an endoplasmic reticulum, that's a mitochondria, that's whatever. If I give you a description of what it does, converts glucose, and the energy for storing glucose into ATP, you need to tell me, oh, that's a mitochondria. It's a very large, fluid, water-filled vesicle in a plant or in an animal. Oh, that's a central vacuole, or it's a vacuole. You need to be able to identify what it looks like and what it does. You also need to know what it's made of, okay? I'll come back to these ideas later, but these are the kind of things that we want to think about. I will create some homework quizzes that will help you to practice some of these ideas, like what's it made out of, or what does it do. I may give you like a picture of a cell and you've got a label. That's going to be a little more just rote memorization, okay? It's putting together lots of these pieces. And then from there, to finish out this semester, we'll get into some of the actual cellular activities. Like, we've talked about metabolism, so we'll look at photosynthesis, we'll look at respiration, and what those are and what those look like. That gets written. I mean, there's just going to be, like, you're just going to have to memorize formula, you're just going to have to memorize pictures, and I'll give them to you. And when I say memorize them, I mean you have to memorize them. There's nothing else, there's nothing to it but to do it. And then we'll finish up with DNA and transcription, translation, DNA technology, stuff like that. And that'll finish out. We'll be at Thanksgiving before you know it, okay? Gotta study your notes. You've gotta review your notes. All the videos and stuff that I show you in class, I put on the Moodle, so you can go watch them again. You gotta read your book. If you will just read your book, read through your notes, come to class, it'll stick. That repetition of hearing these things with a slightly different voice. You know, I talk different than the videos. The book talks different than me. I even put the book's PowerPoint slides up there that you can see. Remember, I don't do everything that's in this, but sometimes I got stuff that you don't need this. You know, it's one more thing to memorize. We don't need to do that. But you've got multiple sources. Avail yourself of them, and it'll get in there, right? It's worth doing. Okay. Well, I want to pick up where we left off last time. So we're going to go here.
Round and sight.
Anybody?
I think we got
What's the last thing in your notes? The last organelle in your notes?
flagella.
Okay. Anybody have anything different?
I don't know if flora plant was after that or not. I don't know. That's not what that's this.
Chloroplasts.
Yeah, chloroplast. Chloroplast. I don't know where that is.
Did we do cilia?
I think we're past
That's this. Well, this is near the end, so I'm just going to hit start here. Okay?
That can move in waves. This feature helps trap inhaled particles in the air and expels them when you cough. Another unique feature in some cells is flagella. Some bacteria have flagella. A flagellum is like a little...
So I'll just tell you this. This would be a classic Douglas question on a test. Name the cell in humans that has a flagella. Sperm, right? Okay. the same question could be asked in a different way. It's a whip-like structure that propels a cell in dozens or hundreds. There's small hair-like structures that work together to propel a cell. What are those? Cilia. Yeah. Okay. All right.
Tail that can help a cell move or propel itself. The only human cell that has a flagellum is a sperm cell. In summary, remember, eukaryotic cells are plant and animal cells with a nucleus and membrane-enclosed organelles, while prokaryotic cells are unicellular organisms without these things. All cells have a cell membrane, cytoplasm, and genetic material. And even though only plant cells have chloroplasts, both plant and animal cells have mitochondria.
I just finished that video last time.
We might have. Okay. Let me back here 1 second. Oh, that's just a, so this is just another resource you can click on. It's just gonna, it's there on the Moodle. It's just gonna show you, an overview of a cell. It's just gonna talk about the same things that we've been talking about. Okay. All right. Well, let's go ahead then. It's working now. Okay, there we go. All right, gang. What I want to do for a minute is catch up and make sure that I'm covering the stuff that's kind of in the basics in the book. So prokaryotic cells, eukaryotic cells. We've talked about this. We did a big diagram. Pro, what does that prefix mean? No, that'd be bio. That's okay. Pro. Proactive. Prohibit, procreate, procrastinate. Pro means before. Okay, before. These are. Remember I told you you're going to learn a lot of Greek in this class. That's what we use in biology. Pro means before. You means good or true. For instance, the gospel comes from this word in Greek, euangelion, good news. Angelion, you are familiar with this, even if you don't know it. It means angel. Well, angelos is angel, messenger. So euangelion is the good message. It's the good news of Jesus Christ dying for your sins. So eu means good or true. So one is before this idea of a karyote, which is referring to the nucleus, and the other one is a true nucleus. So a prokaryote, that's before there was one. There is no nucleus. The other one is a true nucleus, right? So if I just drew two cells, and if you saw nothing else but this, well, you'd go, okay, that is a eukaryote, that is a prokaryote. How do you know? That one has a nucleus, and that one doesn't, right? They both have some genetic material, but there's a difference there too, right? What name a prokaryote that you should know? Simple, unicellular. All prokaryotes are unicellular, by the way. They just don't have enough structure. They don't have enough information to become multicellular and complex. So this is unicellular, right? Name a unicellular prokaryote.
Bacteria?
Bacteria. Amen. Bacteria. Very good. Another one would be archaea. Sometimes you'll see them called archaebacteria. You might not know that you know about these, but you know about these. Anybody ever been to Yellowstone National Park with the hot springs? Have you ever seen pictures of the Grand Prismatic Spring? Okay, just stop what you're doing. I got to show you this. Okay. You've never seen the Grand Prismatic Spring. I got to show you this. This is not colored hands. This is what this joker looks like. Make sure that you can see on this too. There we go. Look at that. So, oh, go back. So the reason this looks the way it does is the water has a lot of copper in it, so it's blue. But all these different colors are archae. They're unicellular do photosynthesis. They do what's called chemosynthesis. They use the chemicals that are coming up out of this hot as an energy source, and they make glucose, and they live off of that. That's crazy cool. And they can survive ordinarily hot or acidic environments. The reason we're talking about them is they're prokaryotes. So bacteria and archae, or bacteria and archaebacteria, these are our two prokaryotes. Let me just tell you right now, you must know that, okay? I'm promising you that will be on a test. You must know these two things, prokaryotes. You must know that they're always unicellular. You must know that. You must know that they're simple. They're very, very simple. What is this little, like, this isn't a nucleus. Notice I drew it, you know, kind of like squiggly, and what is that? It's called DNA. This is their DNA. Their DNA exists in a ring. It's just a simple ring. That's all it is. Coming out of a ring of DNA. Whereas over here, my DNA exists inside of my nucleus, right? And it can exist either as chromatin. I don't expect you to know this yet, but you're going to have to. Or it can exist as chromosomes. It's coming. Chrome means colored, by the way, because we can stain the cell and see the DNA. It'll take a stain, and so it's colored. Chromosome. Chromosome means body. So when they first started doing work with cells and they'd stain them, they'd find these colored bodies in the nucleus. They didn't know what they were made of. They didn't know what they did. They just knew that, wow, that's different. So they called them chromosomes and then chromatin. So this is how DNA exists here, and this is how DNA exists here. Do you know about your chromosomes? Do you know how many you have?
23.
About 23. You have 23 types. But you have 2 of each type. One from your mom, one from your dad, so you have 46. 46 chromosomes. All right, so let me just draw, I don't know. This won't even be even close to accurate, but I'm going to do the best I can. So I'm going to draw like a chromosome, okay? All right, so there's one chromosome, okay? And comparatively, and you have 46 of these, right? Some organisms have more, some organisms have less. And then over here for bacteria, this is their DNA. Well, which one's bigger? Which one's bigger? How much bigger? Thousands of times bigger. And you have 46 of these. That's 46s of thousands of times bigger than that. What's on here? It's made of DNA. What is DNA good for? What does it do? It's going to make RNA, which is going to make proteins. What particular information do we call the information for one protein? We call it a gene. One gene equals one protein, right? This is thousands of genes. 46 is of thousands of genes more than that is. Which means I can make like so many more different proteins than this one can. These things are simple. Hundreds of genes, hundreds of thousands of genes. right? I mean, huge difference in scale of complexity. And yet, these things are way more prolific than you are. They're everywhere. There's nowhere on Earth you can go that you won't find bacteria or archaebacteria. They're everywhere. They're so wildly successful. Sometimes simple is good, right? Anybody drive? You know? Anybody drive a car, whether it's yours, your parents, or whatever, but it's got like all the fancy electronics? Good luck if that joker goes wrong, right? You can't fix that yourself. Get yourself an old Ford truck, though. You can fix that. That's the idea here. Like, you're way more complicated. There's also a lot more to go wrong. These guys, much simpler. They thrive. So, okay, prokaryotic life is always unicellular. It's always simple. It's always a little bit of DNA that exists in that ring. Okay, then there's eukaryotic life. Right? So eukaryotic cells, name a couple things you know every eukaryotic cell has. By definition, what's it have? Has a membrane. What else? A nucleus. What else? What's in between the nucleus and the membrane? What's all in there? True. What's the fluid that's in there? Cytoplasm. Good. You know they're going to have those things, right? So if you see a nucleus, you know that's a eukaryotic cell. And the question is, well, which kind of eukaryotic cell? Well, what kind of eukaryotic cells are there? Plant, animal, and fungi. Good. So how will I recognize if it's a plant? What's it going to have? So wall. Good. So that it just structurally it's going to look like that, you know, or something like that. It's going to be pretty rigid. They don't always look like this. I mean, you look at classic, you look at like onion cells, and they look like kind of like this. They can look different ways, but they're going to be angular, okay? And then inside, I'm going to have, what's that? Nucleus. Or no, that's membrane. Sorry, I will have a nucleus. Let's do a nucleus. But what is one other just dead giveaway? Hey, this is a plant cell. Got the membrane. Got my cytoplasm. They all have cytoplasm. Chloroplasts. Chloroplasts. Now, not every plant, all plants have chloroplasts, but not all plant cells have chloroplasts. Would you expect to see a bunch of chloroplasts in the roots? That wouldn't make sense, right? There's no light to do photosynthesis. You wouldn't expect to see it in the xylem, the part that's inside of the tree. In fact, those cells are dead anyways. Isn't that crazy? You build a cell just to have it die. That's what they do. But in general, if you see a rigid cell wall and chloroplast, you know that's a plant cell. Okay? Yeah, hundreds of thousands of genes. Okay? And then, let's see. If I drew this, what's that? What kind of cell is that? Huh? It's an animal cell. It's definitely eukaryotic. I can see my nucleus. I don't see any cell wall. I could give you more dead giveaways. What if I drew a bunch of these sticking off of it? What are those? Little hair-like structures that move together. Cilia. Yeah. Well, there's some dead giveaways that this is an animal cell. Okay? I've got my rough endoplasmic reticulum. I could have my Golgi apparatus. I could have, you know, I got vesicles. That's an animal cell all day. Okay? And let me draw another cell. Your other type of cell, right? We haven't talked about it much, and we're not going to talk about it too much. Not at this stage. We will later. this looks insane, doesn't it? And then I gotta go put this in everywhere. Looks like some crazy squid done by Picasso or something, doesn't it? I'm gonna put one here, I'm gonna put one here, I'm gonna put one here, put one here, here. What am I drawing right now? We got,
Fungi.
it is a fungal cell, but what did I just draw? Was the last thing I drew? Nuclei. These are polynucleate, many nuclei. One of the reasons they need to be polynucleate is that cell's crazy. It's stretching out everywhere. If I need a protein over here, good luck if I have to get it from over here. Right? And so if I have a nucleus over here, I can control it over here. They're polynucleate. Fungal cells are crazy. They're wild. Anyway, we'll learn more about them later. They have a cell wall, right? They have so those are some dead giveaways, because you better believe I'm just going to give you pictures and be like, hey, what's this? Hey, label this structure. Hey, what does this do? That's how it's going to go. Okay, so if you ask what the test is going to be like, that's going to be on there. All right. What else do you need to know? Prokaryotic cells. Okay, they want you to know more than I gave you. That's my fault. So they gave you this classic picture. So just expect to have this picture and have to label it, okay? Kapish? All right, here we go. Every textbook ever has this picture. It's a nightmare. You're not going to be able to draw this. I'm going to have to go draw this. I'm going to give you this. I'm going to print off one of these for each of you, okay? And you'll have it. It'll be great. Let me do it right now.
I see what you're trying to do now. You're going to draw and then draw the
So what else is in here that I didn't necessarily cover? Covered that. Definitely covered that. Talked about that. That. Definitely did that. That. Maybe we did pretty good. That's good stuff. I know what the book knows. All right. So let's do a couple charts, just comparison charts, okay? Prokaryotic, eukaryotic, then under eukaryotic we're going to break it out to plants, animals, and fungi, okay? I'm just going to work this a little bit. First thing I want to know is, does it have a membrane? Next thing I want to know is, does it have a nucleus? Next thing I want to know is, does it have a flagella? And cilia. And let's go cell wall. Let's keep going across. Filling this out. Should be able to do this without me. That's the goal. Membrane, nucleus, flagella, cilia, cell wall. What else do we need on here? How about a nucleolus? How about a central vacuole? How about a lysosome? Polynucleus. What am I missing? Ribosomes. What else are we missing, gang? Endoplasmic reticulum. Golgi apparatus. Oh, mitochondria, my goodness. Vesicles. We'll stop there. That's fine. All right, help me out. Membrane. Who has it?
Sorry.
Do prokaryotes have a membrane?
No.
If you're going to be a cell, you must have a membrane. Everybody has a membrane. Okay? Who has a nucleus?
Eukaryotic.
All the, okay, eukaryotic, so everything under it, right?
Yes.
Who has a flagellum? Huh?
Prokaryote.
Prokaryotes do? Who else? Well, tell me.
Animals.
Animal cells do, yes.
That should be it, right?
Well, it's actually everybody. What you might not realize is that plants have sperm cells. Fungi have sperm cells. Isn't that crazy? Yeah. So it's not in many, but it's there. Cilia.
Plants. Or I mean animal cells.
Definitely in animal cells. They're in some prokaryotes. Cell wall. Where will I find cell wall? Everywhere except for
Prokaryote.
everybody but animals. Good. What are cell walls good for? Yeah, providing structure, providing support, making it rigid. There are advantages to that. There are also disadvantages, right? It's hard to move. Here's a tree moving. Ready? Oh, come on, y'all. You're giving me my best tree impression. Look up. This is like Academy Award winning tree impression. Ready? Look up. This is a tree running. This is a tree smiling. This is a tree doing jumping jack. Like, they don't move. All right, nucleolus. It's that section inside the nucleus where ribosomes are made. Who has a nucleolus? But you never thought about that. Who do you know has a nucleolus? Plants do. Who else does? Animals. Why don't we just click eukaryotic, okay? Central vacuole. Where will I find that? It's the biggest cell. Plant. In a plant. I guess you could click eukaryotic also. If I gave you something like this, I wouldn't put eukaryotic as a chart. I would take that away. I realize right now that didn't help you. Okay, what's next? Lysosomes. Who has a lysosome?
Are the exes where they go?
Yes, X's happen. Yeah. Who has a lysosome to tear up stuff that you don't want? Who do you know has it? Because I've talked about it a lot. Animals. You've got it, right? Some plants have them. Polynucleate. I've only described one organism as being polynucleate. Fungi. Those ribosomes. What do ribosomes do? They make protein. Who needs to make protein? Everybody. So everybody has ribosomes. Very good. Endoplasmic reticula. Where would I find an endoplasmic reticulum? Where in the cell? What's it attached to? The nucleus. So if I don't have a nucleus, I don't have an endoplasmic reticulum. So it's out on these guys. It's here for everybody else, right? What are the two types of endoplasmic reticulum? Rough and smooth. What's the difference? Rough has? What makes them rough? When you were a kid, or maybe even now, did you ever have like those bedazzling guns? Yeah. So that's like rough ER. It's like bedazzled with ribosomes. Form fits function, right? If I'm going to have ribosomes, well then my function. Okay, Golgi. These modify all these proteins and stuff like that. Do you remember kind of the order of operations that stuff gets made there on the endoplasmic reticulum, and then it forms the vesicle, and that vesicle goes to the Golgi apparatus, and things get modified? So if I have an ER, I probably have a Golgi, right? Just think about that for a second. So who's definitely not going to have a Golgi apparatus? Yeah, definitely prokaryotes. They're not going to have it, right? You know that an animal will. I'm curious if I don't know why they don't list fungus on this. But anyway, so we're going to go here. Mitochondria. What do mitochondria do? Convert the energy that's stored in glucose into the form that cells can use called ATP. They're the powerhouse of the cell. Who has it? Plants have it. Who else? Animals. Fungi. Who doesn't have it? Prokaryotes, right? Vesicles. What are vesicles?
Cold water.
They can't hold water, but they can hold almost anything. They're little sacks. They're like Ziploc bags of membrane inside the cat. So who has them? Who doesn't have them? Prokaryotes. They don't have them. They're made generally either at the cell membrane or at the endoplasmic reticulum. Sometimes at the Golgi also, as things are coming and going. But if I don't have those, I don't have that, right? So, yeah, I mean, look at how much is missing for prokaryotic cells. I'd like to talk about the characteristic of what's missing. I'm going to draw your attention to something here. You've heard this before, but it's okay. So, a nucleus, what's it made of? What's in it? DNA. What's it made of? Genes, and what's it by? Well, that's outside of it. Sorry, so in that way I guess it is surrounded by it, but like a nucleus together, what's inside? What's it made of? Membrane. So the nucleus has membrane. Okay. Central vacuole, what's it made out of? It's mostly water inside, but inside of what? Say membrane. A lysosome, I got a bunch of enzymes inside of? Come on, there's a pattern here. Inside of membrane. Polynucleate, again, the idea of a nucleus. So it's made out of membrane. Endoplasmic reticulum. It's made out of membrane. Golgi apparatus, it's made out of membrane. Mitochondria are surrounded by a membrane. Vesicles are nothing but membrane. And somehow I left off a very important one here. Chloroplasts. Guess what it's surrounded by? Membrane. So other than the nucleolus that's inside of the nucleus, like everything else that's up here that eukaryotic cells but prokaryotic cells don't have is made up of membrane. One of the hallmark differences between a prokaryotic and a eukaryotic cell is the term membrane-bound organelles. Remember, I just described things as being like Ziploc bags. These are like Ziploc bags. You can contain stuff in them. You can separate stuff in them. You know, my little kids are at their co-op school today. On their way to co-op, I'm sure my wife has like pretzels in this Ziploc bag, and she's got carrot sticks in this Ziploc bag. You don't want your pretzels and your carrots in the same bag because then your carrots get dusty and your pretzels get mushy. And who wants mushy pretzels? You want to keep inside things in and outside things out, so you separate them with a membrane because you have a decent mother who's a human being, right? And if you're a cell, you don't want the thing that is responsible for giving you energy getting destroyed by the thing that destroys stuff. You need to keep the destroy things in their own compartment and the energy stuff in their own compartment, and you do that by surrounding them by, say, membrane. By membrane, right? And what's that membrane made up of? There's just a hair of protein, but what's the number one thing I'll find all over the place?
Ribosomes.
A lot of things that I should understand.
Lipid. What's the theme that's been running throughout the entire year so far? 2 words? Charge and shape. You can do this, and you can keep things separated and highly specific and organized and operating like a machine because of the charge and shape of phospholipids. Because they do this on their own every single time they form this phospholipid bilayer. If you just change the charge or the shape of that thing a little bit, you lose that and there's no life. Guys, if there's not membrane, there's not life. If there's not phospholipids, there's not life. You change the charge, the shape, the electronegativity, there's no life. Everything hinges on something as simple as one atom having a greater affinity for electrons than another, or the fact that hydrogen and carbon have nearly the same affinity for electrons as each other. And so they form these polar and non-polar bonds. And so that bonding thing we did the first week is still here in the middle of the semester, and it's still going to be here. You can't get away from it. We talk about animal life later on, and animals adapting to their environment. And I mean, my gosh, you can just talk about how things, like a fish in a pond, in water, like it's got to change in the winter. It's got to change the structure of its cell membranes so that the membranes can flow better in winter, so they don't just freeze up and not function, they die. So you've got to remove cholesterol so that the things flow more readily. But then in the heat, when everything's moving too fast, you don't want your cells just like ripping apart. So you add more cholesterol, hydrophobic, so that it bonds more tightly so things aren't as freely moving. That's an adaptation to the environment and the temperature, and that's ecology, and yet it's still chemistry, and it still are two words, charge and shape. All right. By the way, there's a chart very similar to this in your book. Okay? But this is a good activity. It made you think, right? We need to think. Heavy lifting around here. All right. What else do we have going on? How much time do I have? 30 minutes? Something like that? 25, 30 minutes? Okay, that's good. All right, gang. Well, I think I'm done with this. Okay? You're responsible for everything I've taught you. You're responsible for what's in the book. Go back and review. I'm not coming back at this again, okay? I will give you homework quizzes for it, though. You got to be able to draw it. Write this down. Real big, real big. For my test, I must be able to draw it, or at least know what it looks like on a drawing, right? I got to be able to recognize it, label it. I should probably be writing this down, shouldn't I? Be helpful. Okay, so you must be able to draw / label. Okay, you've got to know its structure. What's it made out of? You've got to know its function. What does it do? You better know what kind of cell it's present in. What is it present in? Is it prokaryotic? Is it eukaryotic? Which types? What else do you need to know? Draw, label, structure, function. What is it present in? That's pretty good. You should be able to compare and contrast. Between prokaryotic and eukaryotic. Between plants, animal, fungus. If you can do those things, you're going to be good. Okay? You're going to do really good. kind of turn the page in some ways, but in other ways we already did a lot of this, because when we talked about membranes, I went hard and heavy into membrane, right? About their structure, about their function, things like that. The next chapter in the book is all about this. So one thing they throw out is this idea of what they call a fluid mosaic model. So our theme net right now is membranes, right? And they throw out this idea of a fluid. What does fluid mean? It does what? It moves. Fluid mosaic. Do you know what a mosaic is? You don't know what a mosaic is?
Wasn't it like when you put a bunch of pieces together and make a picture?
Yeah, to make a picture, right? Like they do it out of tile or marbles or glass or whatever. Some are quite beautiful. Some of them are cheesy and terrible. But fluid mosaic model. So it's fluid, it moves, it's dynamic, it's changing, and it's made out of stuff. What are the mosaic pieces of a membrane? Remember drawing a membrane? What do we draw a lot of? Lots of circles with two squiggles. What are those called? Phospholipids. Okay? So one of the things that we know is lipids. What else is it made out of? I just talked to you about one big one earlier. I talked about like cells and needing to change the composition of their cell membrane. Adding or subtracting this particular steroid, this 4-fused carbon ring hydrophobic lipid molecule that fits very nicely in the middle of a phospholipid bilayer, but it restricts movement. It's called cholesterol. I talked about things needing to be able to get in or out of a cell, and sometimes we pump them, or sometimes we have a channel that they can pass through, and those pumps and those channels are always made out of which type of biomolecule? Say it. Protein. Good. And another one that's used largely with like identifying cell types and signatures is another biomolecule. These are all biomolecules, right? Like lipid, protein. Well, we got 2 more classes. It's either nucleic acid or it's carbohydrates. It's not nucleic acid, so it has to be carbohydrates, right? Okay. So the mosaic aspect of the fluid mosaic model is it's phospholipids, cholesterol, proteins, carbohydrates. Your cell membrane is made up of these, but the background of it all, right? The thing that outnumbers everything is what? Phospholipids. What makes this fluid? What allows it to just flow and move? Which of these? No, that inhibits it. No, which one of these is responsible for making it flowy and fluid and moving? They move very easily and readily next to each other. Phospholipids. Remember, the phospholipid is very similar to an oil. An oil is a triglyceride, 3 fatty acid chains attached to glycerol. This would be a diglyceride, 2 fatty acid chains, and you replace the third one with a phosphate, which has a positive charge. Excuse me, a negative charge. And so you got a hydrophilic and a hydrophobic. These things flow like oil. They flow very easily. So it's a fluid mosaic model. It's important that your cell membrane is not rigid. If it was rigid, it would bust easily. If a cell got a little bigger, a little smaller, it would just burst. You couldn't change. You couldn't get things in and out very easily. But as it is, you can do all of that. Because it's fluid, it's made up of all these parts, hence mosaic and model. You know what a model is. Okay. Oh golly, we've drawn it. We've drawn it. We've talked about and drawn phospholipids. We've done the bilayer. We've seen all that. What else? Selectively permeable. Let's talk about that. That's our next major concept. Okay, I'm going to come over here and erase. So these things are, membranes are selectively permeable. What does the word selectively mean?
You get to choose.
Choosing, right? You're looking for a husband someday, you better be selective. There's a lot of trash out there, right? No Glorilla. All right, here, you better be selective. Permeable, do you know what the word permeable means?
You can move.
Not move,
You can get inside.
it can get inside, it can pass through. Okay, so it's very selective about what can pass through. It's choosy. All right? Some things pass through much easier than others. Hydrophobic things pass through easier than hydrophilic things. All right. What do they want you to know about this? Some substances pass through, but not others. If they were to lose this selectivity, the cell would no longer be able to sustain itself, be destroyed. Materials. Okay. The most direct forms of membrane transport are passive. Passive transport. Passive, because there's two types of transport, okay? Transport across a cell membrane. There are two kinds. One is active, and the other is passive. Describe someone or something that's passive. A person. Describe a person who's active versus a person who's passive.
Dead.
Short of dead.
Like shy.
Okay, maybe shy. What else?
Passive aggressive.
Well, you're not helping me out there very much, ma'am. Describe somebody who's active. They use a lot of energy. Somebody who's passive does not. So this has to do with energy, right? So somebody who's active uses a lot of ATP, which of course is energy. By the way, ATP is almost always drawn like that, with like some kind of an energy bubble around it or something like that. Passive is no ATP. Okay. So one uses lots of ATP, one does not use ATP. One uses energy to get stuff in or out of the cell, the other one does not use energy to get things in or out of the cell. Okay. We've talked about diffusion, right? Okay. So diffusion is a form of passive transport. That's just simply something moving down its concentration gradient. I described that to you before, didn't I? It's moving from high to low concentration. Let me come over here and do a little. What time is it? I don't want to run you out.
10:40.
What time?
10:40.
So I got 10 minutes? Okay. All right, so diffusion. Laugh at me. It's a process of transport. A single substance moves from high concentration to low concentration area until the concentration is equal across a space. Great. So it's movement from high to low concentration. You've heard that a lot, right? You even have drawings of this. And then we talked about, in particular, diffusion of water across a selectively permeable membrane, and we call that the diffusion of water across a selectively permeable membrane is called So then we kind of connected to this, we talked about three terms: hypo, hyper, and isotonic. Tonic referring to the concentration of solutes. Hypo versus hyper. Hyperactive, hypoactive. Which one's more active, hyper or hypo?
Hyper.
Hyper. So this one has higher levels of solutes. This one has lower levels of solutes. Isotonic has same, iso means same levels of, or equal levels of solutes. Once you get that finished, and we're going to draw the next thing. How are you, sir?
I'm okay.
It's a shady bunch, don't you think?
I came to drag her out of here kicking and screaming.
What happened?
Nothing yet.
So hyper, hypo, and isotonic. I'm going to draw something. You tell me which thing is which. If I have a selectively permeable membrane right here, which one is hyper, which one is hypo?
The one on the left is hyper, the one on the right is hypo.
Yeah, this is hyper. This is hypo. Everything wants to follow its concentration gradient. Things want to move until they are iso. So if things are going to follow their concentration gradient, which way are the solutes going to flow? Let's call this into A. Let's call this A and B. Solutes, do they want to flow more into A or B? They want to flow from A into B. Which way does water want to flow, into A or B?
A.
Into A. And water will do that, and salt will do that until they are
Iso.
equal. And keep in mind, they can go back and forth if it's selectively permeable membrane. So, turkey, you guys ready for a Thanksgiving tip? Best one you're ever going to get? You want the juiciest turkey you've ever had in your life, even if you're not the one who prepares it, you just tell your mom this is what you do? Okay. You get yourself the turkey, and you cut it open, and you take the neck out, because that's gross. And the giblets, don't let them make giblet gravy, it's disgusting. Nobody wants that trash. So, do that. You get yourself a big cooler, okay? Like, no, like, yeah. And put your turkey in there. You need to do this in the bathtub. Are you taking notes? This is serious. This is life and science and delicious Thanksgiving. Your mom can't mess this up, okay? You put it in the bathtub, because this thing's huge. How else are you going to fill it? You gotta think, right? You're not doing this in the sink. We just brought an igloo cooler into this situation. Put your turkey in there. Pour a bag of ice in there. Fill it up with water so that your turkey is covered in ice water. Ice is necessarily so that the prokaryotic bacteria don't grow on it and do nasty stuff. You want it to stay room temperature. Another thing that's going to help with killing bacteria is you're about to pour a ton of salt in that thing. You're going to pour salt in like pour a whole, you can't get enough salt in this, okay? Just wait. Remember I said you're going to get the juiciest, tenderest, most just amazing turkey of your life. You're going to pour salt into the water. My salt water is going to be hyper or hypotonic compared to my turkey.
Hypertonic.
Hypertonic. The salt wants to flow down its concentration gradient. Where's the salt going to move? Into the turkey. Water is going to want to move in or out of the turkey. Out of the turkey at first. So it doesn't sound like a very juicy turkey right now, right? It sounds like we just dried our turkey out. But we didn't, because as salt moves in, now water's going to want to move in until the two are isotonic. And so by putting all that salt in, the salt moves into the turkey cells, the water flows into the turkey cells. This will, and in between the cells as well, you are going to have the juiciest turkey you've ever imagined. And you know how like your mom never gets the turkey quite right, it's always like it always needs some seasoning? Not this year, because you got the salt in every cell over the course of about 3 days. It needs at least 2. I guess you could try to rush this over 1 if you had to. There's a reason you're doing the ice, right? Your mom was probably keeping it in the fridge for a couple days anyways to be thawed. So I'm serious here. Do this every year. You get the cooler, you put the turkey in, you fill it with ice water, you pour the salt in. I also like throwing like some thyme, some sage, some.
So you do this?
Every year. Every year.
Do you keep adding ice?
No, you don't need to. You got a good cooler, it'll stay. And then you put it, it's called brining. Okay, that's all this is. Oh, now it's okay. I used the word you knew. I'm telling you the science, and you're like, that's weird. If I say brining, oh, that's cool.
You discovered this.
So, on the Food Network, when I was younger and single and had time, I would watch the Food Network. And Alton Brown had a show called Good Eats, and he would have a Thanksgiving special that would come on some years. And he would talk about doing this, and he would like draw out cells and what was happening with the water and the salt and everything else. It was actually kind of cool and nerdy, right up my alley, and delicious. So I did it that year, and I've done it. Once you've done it once, you never go back. It's so easy. So easy. It's the most, now, if you really want to double down and make it the best turkey of your life, go smoke it, like cut it in half, and go smoke it for an hour, and then finish it in your oven. It's amazing.
You really like turkey, huh?
I really like cooking and science. So we're probably about done, huh? All right, let's finish this. So everything wants to follow its concentration gradient. But what if solutes can't move? What if this is, because it's selectively permeable, what if it's not permeable to my solutes?
So what is the
Water's going to flow into
the
A,
and then it's gonna shrink.
until it expands, right, as much as it can. Now, eventually, it can't hold any more water because there are multiple factors in here. But you don't need to know all that right now. Yeah, but this you gotta know. And they'll do this until it's isotonic, right? Solutes will always flow from hyper into hypo, and water will always flow from hypo into hyper until they're iso, right? They're doing, water and solutes are doing opposite things. Just like that turkey, right? At first, water wants to flow out of the turkey into the salt water. At the same time, the salt's flowing out of the salt water into the turkey. But then eventually the water will flow back into the turkey more than it ever was, and it's going to be, right? Science is delicious. Okay, hyper, hypo, isotonic, osmosis. You've got to be able to regulate all this, right? Otherwise you will die. Okay, tonicity in living systems, don't care about that. Why don't we stop there? We'll pick up with active transport next time. There's not going to be a ton to that, and then we're done with this unit. So, test next time. We'll finish this unit, the class, after, like in 1 week. So we'll probably, we can do this math right now. Let's look at this for when your test is good. Next test is going to be. Okay, because it's coming fast and furious now.
I won't be here next Tuesday.
Okay, well, hold on. So, all right. Test this Thursday. I will finish lecture on the 23rd, no, on the 24th. I will not test you on the 26th. I will test you on my anniversary.
On the Monday?
October 1st.
I mean the Tuesday?
Yes, October 1st. Now I know when my anniversary is. get married, don't ever mess that up. Ever.
Have you missed yourself?
Not by forgetting it, but by not knowing how to be perfect. Yes. You better have a plan. It better be your plan. It better be a good plan. There better be food. Anyway.
Okay, all this that you're talking about today on like stuff today being on test questions, that's for the next
Yeah.
test, not this one, right?
Correct. Yeah. Because I put the first test off so long, because we just kind of drug and drug and drug, then now it's like, okay, we're gonna hit the second one pretty fast. But Lord bless you, keep cause his face to shine upon you. All a great day.
No,
Text me. I will not read it.
I realized that after texting you, I was like,
Yeah, you
oh.
texted me and I was at work.
I was like, I know not to ignore it, and I didn't even know what to do.
No, he didn't work. Probably school. Yeah.